Hello and welcome everyone. In last class, we learned about DNA methylation. In today's class, we'll be learning about DNA methyltransferases. So DNA methyltransferases are the enzymes which will be adding methyl group on the fifth carbon of the cytosine of CpG dinucleotide. Okay, so when the studies about DNA methyltransferases started, scientists got to know that there is an enzyme which transfer methyl group, and they named it as DNMT1. Okay, so initially they thought that DNMT1 is the only enzyme, only DNA methyltransferase which is owned by cell. And they also believed that it does both de novo methylation as well as maintenance methylation. Okay, so let's first know about what is de novo methylation as well as maintenance methylation. So, de novo methylation. De novo means new, while uh, maintenance methylation. There are two types of methylation. Okay, so whenever so has it ever occurred to you that how the identity is maintained? When we were discussing about epigenetic, we discussed about case studies like Dutch hunger winter. I often asked you a question: why do twins differ? Right? Both. Why do the cells which have same DNA sequence differ? So the probable answer that researchers thought that they came up with the hypothesis that cells have the same seat of DNA, but they read the DNA differently to give different outputs. Right? So, have you ever thought that when the cell divides, when a parent cell give rise to two daughter cells or four daughter cells, how do those have the same? How do those have the same output? Like, see, in the twins, both of the cells are gonna have same DNA sequence. Still, the products, the outputs from the cells, the RNA, the transcription patterns would be different. But when a parent cell give rise to two daughter cells, whatever parent cell was producing before will be produced same by the daughter cells also. So why is it so? Because whenever a cell divides, the patterns, the epigenetic marks which decides how the cell is going to read the DNA, they are also transferred to the daughter cells. Okay, just it's the same like your it's the same like as the DNA is passed down to generation, epigenetic marks also are passed down from generation to generation. So this passing down of the epigenetic marks means copying of the epigenetic marks from one generation to other generation, which conserves the pattern of the epigenetic marks. Means you are doing, whatever it was there, you are just copying it from the parent cell to the daughter cell, so you are conserving that epigenetic marks. This is what it's called as maintenance methylation. You are maintaining the methylation marks which was already present in the parent cell. So whenever divides, whenever cell replicates the DNA and it undergoes cell division, the methylation pattern is conserved when DNA is replicated. Okay. So this maintenance of methylation is what we refer as maintenance methylation. Okay. So, but when okay, so this is maintenance methylation. It's just copying of the methylation pattern from the parent cell to daughter cell. Okay. So when a cell undergoes differentiation, okay, like for example, just look at your childhood self and your teen self. Okay, so when you were a child and when you were teen, the identity, your identity is same, but your experiences are different. So like when you were a child, you were kind of immature, but when you grow up, you reach to adolescent stage, you get matured. Why? Because of your experiences. The new experiences which are adding to your life are what decides your maturity, right? In the same way, in the cell, I'm trying to explain this in layman terms. When the cell matures, when the cell differentiates, means it undergoes development through the developmental stages of an individual. When the cell goes from the pluripotency stage to a more differentiated stage, the cell develops new methylation marks, and that is what we call as de novo methylation, means development of new methylation pattern, new methylation pattern when an individual is undergoing development. Okay, this is what we call as de novo methylation. So maintenance methylation is just copying of what there was present earlier, while de novo methylation means addition of new methylation marks. Okay, we are adding new patterns at new stops, at new positions. Okay, so I hope this is clear to you all. So initially, researchers believed that DNMT1 does both of the jobs, de novo methylation as well as maintenance methylation. But here comes the twist. When they observed DNMT1 knockout cells means they do not have DNMT1. Those cells still showed the de novo methylation. Means this proves this hypothesis wrong. Means there is something beyond DNMT1 besides DNMT1 which is doing the de novo methylation. Okay. So for now, this due to this result that due to DNMT knockout one cells which show de novo methylation, researchers now started to explore whether there are other DNA methyltransferases or not. So they performed biochemical assays, to identify whether there are similar proteins or not. But while after a lot of assays, they found nothing. So after doing a lot of biochemical assays, they found no single protein which was doing the methyl transferase activity, which was adding the methyl on the DNA. Then after a few years, when the Human Genome Project continued and other discoveries were there, when the DNA databases expanded, at that point of time, researchers started exploring DNA databases, and they searched for whether there are homologous proteins, means the proteins which share the sequence similarity. And when they explored the DNA databases, and then at that point of time, they found out that there are other members which belong to the methyltransferase family, DNA methyltransferase family, and those members were DNMT2, DNMT3a, DNMT3b. Okay, so at first, DNMT2 was thought, due to its sequence similarity, it was thought that it is also a DNA methyltransferase. But later on, it was discovered that it does do not methylate DNA, but it methylates tRNA. Okay, and that's why it was renomenclated, and it is now named as TRDMT1. So DNMT2 is not a DNA methyl, it is not a DNA methyl transferase, but it methylates tRNA. Okay, so just don't get confused, because it is written DNMT, it is not DNA methyl transferase, but it is RNA methyltransferase. So it is known as TRDMT1, means tRNA, tRNA, aspartic acid. D here stands for aspartic acid, methyltransferase 1. Okay, so DNMT2 or TRDMT1 methylates tRNA. And then the other members, DNMT3a and DNMT3b, they were found that those are the true de novo methyltransferases. Okay, so then scientists established the fact that DNMT1 is actually maintenance methyltransferases. It is maintenance methyltransferase, while DNMT3a and 3b Those are de novo methyltransferases. Okay, so I hope this is clear to you all. So whenever there is copying of the already existing methylation marks, DNMT1 will be playing the role. When there is development of new methylation patterns, then DNMT3a and 3b are working. So there were further there scientists when they developed knockout mice. Okay, when they developed knockout mice for DNMT, each member of this family, DNMT1, DNMT2, DNMT3a and DNMT3b. So they observed those mice and they found out that DNMT1 knockout mice was embryonic lethal. Okay, means the maintenance. Okay, so what happens at embryonic level when the zygote undergoes cleavage, it forms blastomeres. So this the early embryonic stage is all about cell divisions. Okay, right? So for cells to maintain their cellular identity, the already existing patterns should be copied, right? And that's when there is no DNMT1, the cells fail to do that. They won't be able to copy the methylation patterns, and that's why the embryo and that increases the fatality in the embryo, and that's why the DNMT1 knockout mice, they were embryonic lethal. It shows that DNMT1 does the maintenance methylation at early embryonic stage. Just a second. Okay, it does it at early embryonic stage. Okay, so when they studied DNMT2 knockout mice, there was no visible effect. Okay, why? Because obviously it doesn't do DNA methylation, but it actually methylates tRNA. Okay, so this validates this validates the idea that it is RNA methyltransferase, right? So when they observed DNMT3a knockout mice, they showed prenatal death. Okay, means the mice, they were dead sooner or later after the birth. Means they do not survive longer. They were either died before the birth or sooner or later after the birth. Okay, so prenatal death was seen in DNMT3a knockout mice. This shows that DNMT3a is very essential for developing the new methylation patterns in the latter stages of development, not in the early embryonic stage, but it does the de novo methylation in latter stages of development. Okay, while DNMT3b knockout mice, they were also embryonic lethal. Means they played a very essential role in de novo methylation, which is required at early embryonic development for cells to undergo the new to cells turn to a very new leaf. So they do the de novo methylation at embryonic stage, early embryonic stage. Okay, so this was it about DNMTs. So these are the different members of the DNMT families, where DNMT2 is not a DNA methyltransferase. Okay, so just keep it into the take home messages. DNMT1 is DNMT is the maintenance methyltransferase. 3A and 3B, those are de novo methyltransferase, and DNMT2, this is the tRNA methyltransferase. Okay, so now let's take a look at where the DNA methylation is present. So I have already told you that for methylation to take place, there should be the presence of CpG dinucleotide. Okay, so there should be a cytosine which is preceded by guanine. There should be CpG dinucleotides. Okay, so when I say here, cell, the genome, in the overall genome, you would see CpG island, a lot of genes, a lot of repeat regions. Okay, so let's see where are the where the DNA methylation occurs. So DNA methylation occurs whenever there are CPG clusters. Okay, but whenever those CPG clusters have poor CPG frequency, okay, only if you remember the CPG poor island were frequently methylated, while CPG rich islands were infrequently methylated. Okay, so the CPG cluster which has poor CPG frequency are methylated, while the CPG rich islands are not methylated. So when I say here that CPG islands, when I ask whether the CPG islands are methylated or not, I mean here that CPG rich islands. Okay, by the convention, it is thought that CPG islands are they have the high CPG frequency. So the CPG rich islands, they will not be methylated. They will be, okay, they will be unmethylated. Why? Because those are often found at the promoters of the, so where are poor CPG rich clusters are found, sorry, poor CPG cluster, like CPG where the CPG frequency is less, those clusters are found at tissue specific genes, right, for having a better control, while the CPG rich islands are found at promoters of housekeeping genes. Right? So if the promoters of housekeeping genes are methylated, means the CPG rich clusters are methylated, they will silence the housekeeping gene. And if you silence the housekeeping gene, how do you expect the cell to function normally? Of course, so it will lead to the cell suicide, okay, right? If you don't have the housekeeping genes functioning normally, cell will die, and we don't want that, right? And that's why CPG rich islands or CPG islands by convention just get at that those are clusters with high CPG frequency. So CPG islands are unmethylated in the genome. So when while the intergenic regions means the regions between the two genes, those regions are, they are methylated. Why? Because these regions are often non-coding. So you don't want the non-coding regions, the regions those do not they the regions which do not produce proteins, those are known as non-coding regions. So you don't want the non-coding regions to be transcribed. Okay, why? Because you want to you want to save your money. Okay, or you don't want to do the expenses, more expenses, right? So you want to save the energy of the cell, and that's why those intergenic regions are methylated. Why? Because methylation represses the gene. With methylation do not compacts the chromatin structure, and it won't let the transcriptional machinery to access that intergenic region, and the intergenic regions won't be transcribed. Okay, so intergenic regions are methylated, while the repeat regions like transposons, those are also methylated. The reason of methylation of repeat regions is that repeat regions are, if they get active, and they get start transcribing, they will mobilize, and due to mobilization of those transposons, then there will be mutations in the, the cell functionality will not, cell will not function properly, and that's why you methylate the repeat regions so that you can silence the repeat regions. That's why CPG islands are unmethylated. Why? Because you don't want to shut down the housekeeping gene, but you want to shut down transposons, as well as you want to shut down the non-coding regions. Intergenic regions, that's why intergenic regions are methylated, as well as repeat regions. You don't want to mobilize the transposons, and that's why repeat regions are also methylated. So that's it for today. Let's meet in the next class and we'll discuss about the DNA demethylation. Till then, see you. Bye bye.